Good morning, ladies and gentlemen. I am Gonghua Li from China. I was also a visiting scholar of Massachusetts General Hospital, Harvard Medical School. I'd like to thank the organizers for this opportunity to present our recent work. The title of my presentation is Targeting the Host Response in COVID-19 by Integration of Metabolic Modeling and Cheminformatics. This work was supported by Harvard Medical School and the Chinese Academy of Sciences. First of all, let's introduce our team. This is me, this is Fei Fei, and this is the Wenzhong. My work focuses on develop methods for metabolic modeling and the applications to study human health and diseases, including aging, infection, ME/CFS, and neurological diseases by integrating the multi-omics data. In addition, we also intend to develop novel methods for drug design. My presentation contains 7 parts. They are ranging from novel metabolic modeling method to model the viral host interaction and to perform the drug repurposing for anti-COVID-19. As we will learn, virals, of course, including the SARS-CoV-2 viral, usually hijacked the host cell metabolism to support their replication. This process involves high allergy expenditure and a large number of host metabolites. With those hypotheses that modulation of the host cell metabolism may have the ability to control the COVID-19 infection. In this study, we will present a novel protocol to perform the metabolic modeling of viral host interaction, and importantly, we will find novel drug target nutritional supports and repurposing drugs for COVID-19. First of all, we need a method of metabolic modeling. However, the existing methods are currently suffering from a bottleneck that most of them use the quantitative data to construct the fluxes and therefore difficult to quantitatively integrate the omics data into metabolic modeling. In this process, most of quantitative information was lost and may result to fail to reliably predict metabolic fluxes. Our aim is to develop a novel method to quantitatively integrate omics into metabolic models, especially the RNA sequencing data, and to integrate enzymatic kinetics parameters to cellular metabolic models. And then we will want to construct and perform the personalized tissue-specific metabolic modeling. Therefore, we designed GPMM because of the binary nature of the quantitative data. The upper bound in existing methods are either 0 or infinity. Our strategy of GPMM is to quantitatively integrate the multi-omics data and the continuous upper bound for each reaction. We can look at this flowchart. The overall flowchart is to collect the uptake information, the omics data integration, tissue-specific modeling with construction, and the MC sampling and knockout analysis. In order to achieve the quantitative integration, we must solve 2 challenging technologies. The first one is to estimate the protein abundance from gene expression using a mathematical model. The other one is to construct the upper bound for each reaction by integrating the enzyme kinetics into metabolic modeling by using multiplying the K-cut value by the enzyme abundance. By the way, the K-cut values are created from enzyme kinetics database, for example, the BRANDA, and the enzyme concentrations were derived from gene expression data. We performed the benchmark analysis of GPMM. We derived the experimental measured flux dataset of NCI 60 cell lines. After we moved the low accuracy fluxes, we constructed a benchmark dataset contains over 1,000 of uptakes and 500 of circulation fluxes. And then we performed the robust analysis and compared with other 4 popular used methods, including the GIMME, fast co-omics, and EC model. The Weber effect is the most important feature of cancer cell line. We firstly performed the benchmark analysis and studied the Weber effect in NCI 60 cell lines. We can see that the GIMME fails to predict the Weber effect, and our fast co-omics got the best performance in this existing methods with the R-score of 0.33. For GPMM, we observed a dramatically improved performance compared with the existing method. It had an R-score of 0.86, indicating that the GPMM precisely predicted the lactic circulation that present the Weber effect. Overall benchmarking results showed that only GPMM has the ability to quantitatively integrate multi-omics into metabolic modeling, especially the transcriptome data. Importantly, these results showed that GPMM accurately captured the experimental measured fluxes and significantly outperformed the existing methods. For instance, in overall, the existing methods have the R-score of smaller than 0.5, but our GPMM obtained R-score of 0.72. In predicting the lactate circulation, GPMM also dramatically outperformed the existing methods, where we obtained R-score of 8.86. These results clearly showed that GPMM is an accurate method to perform metabolic modeling. Now, the GPMM have been released in GitHub. After solving the metabolic modeling issue, we then focus on developing well-host metabolic modeling protocol. We named it as VHMM. We aim to find useful drug-targeted drugs for anti-COVID-19 using VHMM. We can see that VHMM contains 3 distinct steps. The first step is to integrate viral component from CAMDA map and host metabolic models to reconstruct a host-viral interaction model. The second step is to perform metabolic modeling using GPMM to find the potential drug target and useful nutritional supports for antiviral. For the third step, we will conduct drug repurposing based on the nutritional supports using the cheminformatics. We applied VHMM to study the COVID-19 and performed metabolic modeling to get the host response in COVID-19. We firstly derived COVID-19 components from the CAMDA map, including viral proteins, nucleotides, and lipids, and then we constructed a viral biomass reaction to simulate the viral land. After integrating the viral components and biomass into the host metabolic map, we thus constructed a complex viral host interaction model. This model contains over 10,000 of host reactions and 36 COVID-19 vaccines. Next, we used GPMM to perform metabolic modeling. The input is very easy to obtain and just contain two components. One is the gene expression data. Here we used a dataset of before and after infected syringe cell lines, including the ACE2-induced A549 line, the NHBE and color syringe cell lines. The other one is the nutrition uptake from literature. For the output, it contains fluxes for each sample. In this study, we obtained 3,900 of fluxes from host and 36 fluxes from COVID-19. Besides the fluxes, we also got all against all gene knockout matrix and the all against all metabolite knockout matrix. Using our recently published package FastMM. From the modeling results, we observed that the VHM correctly predicted the viral load. We can find that the RHO is 0.05 between the predicted biomass and the experimental measured viral load. This result indicated that VHM can effectively model the viral host flux change after infection. We also obtained the flux change after infection in syringe cell types. The result showed that most of changes are cell specific. In the meanwhile, we also obtained some common fluxes in these syringe cell types. Next, we look into the pathway changes to obtain the overall metabolic feature after infection. As we expected, the major component of COVID-19 was commonly upregulated among syringe cell types. In the meanwhile, we also found that the mitochondrial transport, glycolysis, folate metabolism, processional transport, oxidative phosphorylation are all downregulated among syringe cell types. These results indicated that COVID-19 hijacked and inhibited the host mitochondrial function to support the COVID-19 protein synthesis. Of course, we also observed the impaired glucose metabolism after COVID-19 infection. We then performed the all against the wall knockout analysis to identify drug target and nutritional supports. We can see here is the all against the wall gene knockout matrix GKO. X-axis is the reaction and the y-axis is the gene. The value in matrix is the ratio of changes after knockout. Zero means not change, while the negative means the gene knockout inhibiting the fluxes. In practice, GKO is the zero or negative. Knockout genes will reduce the fluxes of some related reactions, but if we just look at this matrix, we don't know if these reactions are changed after COVID-19 infection. Therefore, we integrate the gene knockout matrix with change the fluxes and transfer the gene knockout matrix to the gene knockout effective matrix GEM using this formula. The GEM is the multiplying of GKO by the sign of the log 2 for the change after infection, that is 1 or the -1, and by the logic of significance L, the L is can be 0 or 1. Therefore, the GEM matrix has two directions. The negative represents the represent the knockout this gene will rescue the COVID-19 flux change. For instance, there's location of the COVID-19 components. For the positive values, we should consider to activate these genes to anti-COVID-19. As knocking out a single gene can affect multiple reactions, we summarize its knockout effectiveness, and then we will obtain two different types of drug target, pulse drug target and neg drug target. Pulse and neg represent the activation and inhibition of these genes will rescue the COVID-19 host response, respectively. In this study, as COVID-19 may infect various types of cells, we think that consensus targets among different types of cell types are the co-candidate targets for COVID-19, with those overlap the predicted targets among these three types of cell lines. Unfortunately, we don't get any overlapped pulse targets, but fortunately, we obtained 5 neg targets. Interestingly, these overlapped neg targets are all the essential enzymes for purine synthesis. As we well know, purine synthesis is the essential step for viral production. These results suggest that our drug target identification strategy is reasonable. Same as gene knockout analysis, we also transformed the all against all metabolite knockout matrix MKO to the effective matrix MEM to find metabolites the targets. Here the x-axis is the reaction and the y-axis is the metabolites. We can see that there are more metabolites in the metabolic knockout effective matrix than the gene effective matrix. Using the same way as gene knockout analysis, we obtained two different types of metabolites, pulse and neg. Only pulse metabolites can be used for nutritional supports. Indeed, we found 4 overlapped pulse and endogenous metabolites that can be used for nutritional supports, including coenzyme A, NAD+. In the meanwhile, we also obtained 23 neg metabolites that should be reduced in COVID-19. This is not the nutrition, but we could be used for drug repurposing in the next step. After we moved the inorganic molecules, we obtained the full list of anti-COVID-19 endogenous metabolites. The green colored molecules of, for example, the coenzyme A and the NAD+ could be used for nutritional supports. The others, including the glycine, GDP, IMP, are just used for drug repurposing. We hope this list could be further validated by experimental or clinical trial. In the next step, we will perform the drug repurposing using cheminformatics. We have obtained the neg and the pulse metabolites for anti-COVID-19. For example, the NAD+ is the pulse metabolite, and the IMP is the neg metabolites. However, these endogenous metabolites may not druggable and not enough efficiency for anti-COVID-19. Therefore, we perform the drug repurposing to find more efficient and more potent drugs for treatment COVID-19. As the metabolites or drugs act from based on the network or signaling pathway and pharmacological actions, Therefore, we use the three types of feature of molecules, including the network, signaling, and pharmacological action to perform the drug repurposing. As the identified endogenous metabolites have two types, the repurposing strategy should also have two types. For the post-metabolites, we will find drugs that not only have similar network or signaling pathways features, but also have the similar pharmacological actions. For like metabolites, we will find drugs that have similar network or signaling features, but have the opposite pharmacological actions to perform drug repurposing. How to perform drug repurposing using cheminformatics? We firstly used a state-of-art molecular feature database, namely chemical checker or CC, to pre-calculate the correlation between two molecules. And then for each molecule, we fit the correlation using the generalized extreme value distribution and obtained the location, scale, and the sharp parameters for this distribution. All this time-causing process have been pre-calculated. Then, for given any candidate endogenous metabolites, for example, NAD+, we untimely calculated the Pearson correlation features between drug bank and the metabolites, and then calculate the VHMM's network score, and finally rank the repurposing drugs. Next, I will discuss more on this approach. First is about the chemical checker dataset. It is a state-of-art chemical feature dataset, contains about 800,000 molecules. Each molecule has 25 types of signatures. In this study, we use the three types of signatures to perform drug repurposing. It contains the B1 mechanism of action, C2 small molecule pathways, and the C3 signaling pathway. In addition, we also used the molecular structure signatures. For example, the A1 and A2, the 2D and 3D fingerprints to calculate the structure similarity. For each signature, for example, the NAD+, each signature is a vector with 128 elements. For example, the NAD plus B1 and NAD plus C2 is like this. Second, we will calculate the Pearson correlation matrix Cij for each signature. For example, the B1 Pearson correlation between the NADP plus and the NAD plus is 0.78. For NAD plus itself, the B1 correlation between the NAD plus and other molecules is a vector with 800,000 elements. Here is the distribution. Third, as each molecule has about 800,000 correlations, and we have 800,000 molecules in CC dataset. Therefore, the memory usage for calculation is already huge and could be less efficient for the next step. We therefore choose not use the empirical distribution and fit the correlation using the generalized extreme value distribution and obtain and save the parameters. For example, the NAD+, we obtained 3 parameters of GUV distribution of B1 signature of 0.05, 0.11, and 0.15. After calculating the distribution parameters for each molecule, we thus can calculate the similarity p-value between the given molecules and the CC database. If the RHO is smaller than 0, we can use the left tail p-value. This represents the opposite between the query and the dataset. For the RHO larger than 0, we use the right tail p-value. We then calculate the VHMM network score using the multiplying the discriminant factor alpha by log these p-values. In this study, we using the B1 and C2 or 3 features to calculate the score. And the discriminant factor alpha was defined as if both PB1 and PC2 or C3 are smaller than a peak cutoff. For example, the point of 1 alpha is equal to 1, else alpha is equal to 0. Now we can use the VHMM network score to do drug repurposing. We collected the benchmarking dataset from Waveron's Nature paper to evaluate the VHMM repurposing effective. We can see that the repurposing enrichment Ford rate of VHMM reached to 6.58 and outperformed the CMAP. In addition, we also find optimized cutoff using the p-value of 0.01. Using the optimized cutoff, we just predicted 14 anti-COVID-19 drugs from the drug bank. Strictly, we found that the 10 of 14 predicted drugs have known studies and antiviral activities, for example, anti-HIV, anti-SIV, and anti-flu. This result indicates that our method can reliably to predict antiviral drugs, of course, including the anti-COVID-19 drugs. Here are the structures for each predicted drugs. We expected some of them can be further validated by in vitro or in vivo test. For the in vitro test, as our strategy is to modulate the host response to anti-COVID-19. Therefore, there will be some different from the current popular method in vitro drug screen, where the screen the percentage of infected cell. Instead, these predicted drugs should be tested by measuring the viral load after infection. Of course, the ideal testing is the animal model. In summary, in this study, we developed a protocol named VHMM for viral host metabolic modeling and drug design, and we also successfully applied it to study the mechanism of COVID-19 and drug repurposing. We captured the changes in the metabolism of host cells after COVID-19 infection, including impaired mitochondrial function and glucose metabolism. We also identified the potential antiviral targets, including 5 gene targets and 4 metabolites. Finally, we predicted potential drugs for repurposing in COVID-19. Strictly, 10 of 14 drug candidates have known studies and antiviral activities. Of course, this study contains some limitations. For example, the signaling pathway was not included in our models, and we just used, and secondly, we just used the in vitro cell line database. Therefore, in the next step, we will include the signaling pathways and use the in vivo datasets in the viral host metabolic modeling to anti-COVID-19. That's the end of my presentation. Thank you very much. If you have any questions or comments, please don't hesitate to contact us, either to me or Wenzhong. Thank you again for your attention.